crowd tonight. Thank you so much for being here tonight. This is such an exciting evening for me personally. This is, I can't believe, the fourth of our third year. And so it's an honor for me to be able to take people from our FiRe conference that we think are just amazing people, bring them here, introduce them to you, and see what happens. I thank you for putting up with the video again, for all those of you that have been here like every time for watching it. It's really cool for us, though, to finally have brought the FiRe conference to Park City. We've had it at Montage Laguna Beach, and now, for the first time, we had it in October at Stein's, and we're having it again this September 27th to 9th at Stein Erson's Lodge. And at FiRe, we always bring technology leaders together, create solutions to global challenges. Tonight, I'm very, very excited about somebody that I met. I went to the Salk Institute as a gift to a conference, and I love this stuff. I love technology. I love what's happening in the future of healthcare, and we were all kind of led through all these different areas, and we went to the stem cell research lab, and this woman came out to meet us, and she was talking like this about the job, and she was saying, and then we get to do this, and then we get to do this, and then and we could hardly even get in the door, and so I got to walk with Dr. Leah Boyer, and she was showing us what they're working on with neurodegenerative diseases, with organ replacements, and I couldn't get enough of it. So the rest of the team left, and I stayed, and I said, Leah, can I just spend some time with you? And we spent about 45 minutes, and she showed me things that were beating in Petri dishes, and she talked to me about the future of cures of everything that I've ever been afraid of. And I said, Leah, I said, may I have you, can I bring you to our FiRe conference? And she said, don't you want my boss? And I said, no. I want you. And so I'm honored. We had Leah was on a panel at FiRe this last October, and she had her 5 minutes of the 5 people on stage, and I said, that's not enough. So I'm so thrilled to bring her here tonight. I want to tell you about her. So tonight is called The Cells in Medicine, From Protecting the Brain to Regrowing It. That's what Leah works on. Dr. Boyer has been at the Salk Institute working on stem cell research, the head of the core lab for the last 8 years. She studied at Harvard Institute. She studied at Woods Hole. She has a passion for curing Parkinson's disease, and she's brilliant at what she does. She's one of the most brilliant women I've ever met, but the most passionate about her work. So I'm very honored to introduce Dr. Leah Boyer.
Thank you, Sharon.
And I asked Leah, because she's so into this, I asked her to talk slowly and to dumb it down for me, because there's so many things, so many words that I would equate to something that I understand that they use in the labs that are something totally different.
And I told Sharon she had to have like a code for me, so I would
right.
I was talking too fast. Yeah.
So the first thing is, let's talk about what is a stem cell.
Yeah, that's a good place to start. The most basic definition of a stem cell, maybe 6 words or less, is a cell that can self-renew, so make more of itself, and it can differentiate, so it can turn into different things. And that's all it is. And so I thought that the best way to show you guys that would be to do it with a picture. And the picture that I chose is one from 1956, which is before we even really knew that stem cells existed. And it's drawn by a biologist named Waddington. This was back when biologists were also artists, before we thought that those two were somehow mutually exclusive. And what he was saying is that development is kind of like a marble rolling down a slope. And then it's going to kind of go down whatever pathway is the easiest, least resistant kind of pathway for it to go on, and kind of make fate decisions along the way that will then determine what cell type it becomes. And so what we can see here is this cell on top is going to roll down this hill, and then here can make a decision, here can make a decision, and then in the end will reside in one of these grooves. And each one of those grooves is representative of a cell that lives in your body. So you have 100 trillion cells in your body, and they roughly fall into 220 categories. And all I can tell you about that number is that I'm sure it's not right. We're around there maybe, but there's so much diversity within your body. And the way that we pull this off is because of stem cells. Stem cells are what create this diversity. So there's kind of 2 flavors of stem cells. We divide stem cells based on their potential, all the things that they can turn into. And so in this diagram, this right here would kind of represent the pluripotent stem cell, or the embryonic stem cell, as you might have heard of it, that can become anything. Whereas after it's gone down here, let's say it was resting right there, it would still be considered a stem cell because it can still become other things, but it wouldn't be pluripotent. It couldn't become absolutely everything. It could only become a certain number of things, and so its potential is a little bit restricted. And so we would call that multipotent. You guys have probably heard of it as an adult stem cell. And those would be ones that are currently living in your body now, like in your intestine. Actually, the brain is my favorite example to give because a guy at the Salk discovered that there's new neurons being born in your brain throughout your entire adult life. And importantly, the area where these new neurons are being born is called the hippocampus. And that's a part of the brain that's incredibly important for learning and memory. And it's also the part of the brain that gets junked up a little bit in Alzheimer's disease. Right here. So another example would be maybe the hematopoietic stem cell, the one that lives in your bone marrow now. And that's the cell that is responsible for giving rise to all of the different blood cells of your immune system. And so that cell, that hematopoietic stem cell, can differentiate into all those other kinds of blood cells, red blood cells, white blood cells, the T cells, the B cells, the natural killer cells. When we discover cells, we're allowed to name them ourselves, which is why we have cells called natural killers. We have a protein called Sonic the Hedgehog too. So as a kid of the 90s, I'm very excited about that. So those cells, that hematopoietic cell can become anything, but it can't become a neural cell, right? So it's restricted in its potential. It can't really go from right here, this blood stem cell, to right here, down here, without a lot of energy, a really big push. Unlike this pluripotent cell, which can become absolutely everything. And that's kind of where the controversy comes in.
Talk about the controversy of pluripotency embryonic stem cells.
Here's the controversy. This is an embryo. This is a developing embryo. This is about 5 days after fertilization. And to get embryonic stem cells, which are represented right here, we have to destroy this embryo. And that is where the controversy comes. Simple as that. And it's something that I've spent a lot of time thinking about. And I've come to the conclusion that I actually don't have any ethical problems with the work that we do when it comes to destroying these cells. And I'd be happy to kind of take you through that process for me. I kind of realized that, well, first of all, stem cells grow like cancer. They have undivided cell growth. And so once these embryonic stem cells are isolated, they can be cryogenically preserved, they can be frozen and sent around the country. And so at the Salk Institute, we do a lot of embryonic stem cell research. We have about a dozen embryonic stem cell lines, and we have never destroyed an embryo because we don't have to. It's expensive, it's time-consuming, and why would we if we don't have to? If we're having lines from all over the country shipped to us, I often joke that our embryonic stem cell lines don't come from embryos, they come from FedEx, because it is quite literally how they arrive. So that aside, the fact that it happens incredibly infrequently, there's this dichotomy of the moral ethics of embryo destruction in our country that just makes no sense to me. As someone who studied philosophy and took some moral ethics classes, we create these by the thousands on a daily basis.
When you say we?
We as a country. And we're doing this in vitro fertilization clinics, IVF clinics. As a society, we've decided that it's okay to make these if we're doing it for reproductive purposes, to have children that look like us. And I'm okay with that, but we have to understand that every time we go through that process, we generate, maybe a dozen to 2 dozen of these embryos. And we usually only implant maybe a half dozen of them max. And so couples are given the choice to freeze their embryos, and they do so with this idea that they're preserving them, but they're not. Over time, they will degrade, and most of those embryos will never see the light of day, or the darkness of womb, I don't know. Sorry. And so we're destroying these on a daily basis for reproductive purpose en masse. And so to me, someone who watched their grandmothers suffer for 30 years with Parkinson's disease, you can't tell me that it matters more to not destroy 1 or 2 of these, if it could help someone like her not be suffering for decades, that could possibly be not as important as reproduction and creating more babies, and that's an okay reason to destroy them. And I hope that makes sense. To me, the potential value to end human suffering is much greater than the suffering that is caused by this. And the fact that our society does this on a daily basis for reproductive purposes makes me feel like we really don't have any place to say that embryonic stem cell research is not ethical.
And also, you were talking about when somebody's actually getting pregnant anyway.
Yeah, that's true. That most of us don't realize. So this right here is about day 5 after conception. It's hard to say exactly, but 80 to 90% of these will not implant. It's a hard thing. We don't really have a camera in there. We can't really say, but reproduction development, it is difficult. We're not that great at it, but what we're really good at is editing. And our body can tell if things aren't quite perfect. And if things aren't quite perfect, it won't implant. And so most of these most human fertilization events don't implant. And so we're destroying something that might not have ever gone on to develop anyways. And it's just something that, it's a good perspective to have. It's something that's important to keep in mind.
Cool. So let's talk about what your passion is, which is neurodegenerative diseases such as Parkinson's, Alzheimer's, ALS, Huntington's disease. Where can stem cell technology assist us with? They don't use the word cure.
We don't like cure.
They don't use the word cure. We do. They use the word treatment.
Right, because we might not be able to cure, but we don't care about curing. What we care about is making a patient's life better. We want to be able to treat a disease, and if they still have the disease, as long as they're not feeling the symptoms of it, that's all we care about. So I got into stem cells mostly because of my love for the philosophy behind it and the political, but also it was the promise. Stem cells have revolutionized research, and it's because, mostly for neurological disease, because before stem cells, there was literally no way to study human neurons in a dish. Everything that we knew about neurons, about brain cells, about disease, was based on rodent models or other animal models. And rodent models are animals. One of the big things that makes us different from animals, I think, is our brain and how we function. And it's also very difficult when you're working with rodents with brain diseases to really figure out what's happening. You can't really how you doing today? How you feeling? You hearing the voices? Yeah, no, good. You happy? Great, good. Have some kibble. They can't self-report the way that we do. And so most of the drugs that we find in animal models for the brain tend to fail at staggering rates. And so stem cells have allowed us to have these neurons in a dish to really be able to understand human development, to be able to understand human disease, and then hopefully, as I'll show you later, a screening platform for new drugs that we hope will have higher rates of FDA success. But if you think about it, if someone has cancer, no offense to any cancer biologists out there, but it's a little bit more straightforward. No one wants their tumor, right? So we can remove the tumor, we dissociate it, we look at those cells, we see which signaling pathways are disrupted. We can find drugs that target those signaling pathways. But for a neurological disease, we can't really go up to someone and say, so, that's your brain. You still attached to that? Can I have some of that? No, you can't take someone's brain. And so by the time that we actually get to see what's going on in the brain, it's postmortem samples. It's after the patient has passed away. And you can't really tell a lot from a postmortem sample because the disease has really run its course and because the patient has likely undergone treatment for 10, 20, 30 years. And so it's hard for us to say what is a result of the disease and then what is a result of chronic treatment. And so it's one of my PhD advisors, he has this great metaphor. He says that trying to understand a disease by looking at a postmortem brain is kind of like trying to understand why a plane crashed by looking at the debris. You can see why, you can look at all the places, the fuselage all over and try to kind of guess what might have happened, but unless you have the black box that actually kind of records the going down, you're not going to be able to understand it. And stem cells they serve as this black box. They allow us to recreate the disease in a dish and then really be able to understand what's going on. So before 1997, we couldn't do this. In 1997, a researcher from Wisconsin took this little chunk right here, he dissociated it out, and he grew it in a dish. And this is what it looks like. This is an embryonic stem cell colony filled with maybe about 100 cells. So each one of these little circles here is a cell. And what I want you to take away from this, and it'll make more sense when we see some other pictures, that these cells have no cytoplasm. They just have a big nucleus. They have no cytoplasm because there's nothing really that they need to be doing. They're really just here, poised, ready to go. They're like, I'm just nucleus. Tell me what you want me to be. What do you want me to be? And it kind of makes sense if we look at the picture before. They're in this tight little ball right here. They're just kind of waiting. And so this is the morphology that we like to see. It's also the morphology that I wrote my grad school essay on to get into grad school. And the first line was, I see stem cell colonies everywhere. Greasy pizza, cobblestones in the sidewalk. That's differentiation. It doesn't kind of like greasy pizza. I don't know either way. So I spent a lot of time staring at these little guys and then also trying to turn them into neurons. And there's a couple different ways that we can apply them to the study of neurological disease. The first way is kind of what we traditionally think about, replacing cells. And this is, organ farms, or if someone has any kind of degeneration anywhere in their body, that we can replace that which is missing. And that is happening, absolutely, and we could talk about it maybe a little bit later, but for the brain, that's particularly difficult. It's challenging because unlike a lot of the other organs, the brain isn't just these little round cells in one ball. It's highly, the cells of the brain are highly polarized. It's a cell body and then this long axon and these dendrites. The neurons in your brain, they're all different shapes and sizes, and they develop in this kind of very orchestrated, beautiful kind of symphony of just tangles. And so if one little part dies out, it's very challenging to then stick cells back in and expect it to be able to integrate back in then in that same way and then contribute the way that the cells would have had they been in the brain for the whole life. People are still doing this work. There's a trial that will be starting in 2017 in New York for Parkinson's, if everything keeps going according to plan, for transplantation. And there was actually transplantation studies done in the 90s with fetal tissue, and I'm not sure if anybody has heard about these, but they were promising, and then they were also considered to be not worth it enough to the patients to continue, and so they kind of fell out of favor until recently. Some fetal trials are starting back up again in Great Britain, and we'll have the stem cell based trials starting here the next year or so. And I think the stem cell trials, I think they'll be more successful than the fetal trials because the cells that we're putting in are more purified. When you're taking them from fetal cells, it's kind of a little bit more of a gamish. And so hopefully those results will be a little bit better. But our number one thing is do no harm. And for Parkinson's disease, unlike Alzheimer's and Huntington's and ALS, we have a relatively good gold standard of care, levodopa, being able to replace the dopamine. So let's take a step back. In Parkinson's disease, the cells that are missing are the cells of the substantia nigra, which are, hanging out right back here. And they project all the way up to the motor cortex, so that the cell body is here, but the neurotransmitter that's released is released up here in the motor cortex. And so if we lose that transmitter, dopamine, we don't have as much control over what's happening in our motor cortex. And so if we think about the challenges for transplantation, if you want to put the cells back, where do you put them? Do you put them here where they're missing? Do you put them here where we need them to be? So the clinicians, the ones that were doing the fetal transplants, they decided to put them up here where we actually need the dopamine, which makes sense. You couldn't expect them to project up. And one of the problems with the trial was that the patients that had the best results also almost had too good of results because they were moving too much. They couldn't control because the on and off signals, the signals for when to make dopamine and how much to make, those were still going in down here, and these cells couldn't hear them. So it's something that we really do have to kind of pin down, really, really dial in to make sure that we're, if someone has a pill that they can take in a relatively helpful way, to then say, actually, let's cut into your brain. We're putting patients at risk there, and so we really want to make sure that we have that done right. The other way that stem cells can apply to neurological disease is what I do mostly, and that's called disease modeling, kind of creating diseases in a dish. And the way that we do that is pretty cool. So I want to talk a little bit about reprogramming technology, which is a very, very cool technology. Can I see us by hands who's heard of reprogramming? Okay, you guys were here this morning. It doesn't count. Okay, this is really exciting for me then. I think this is so cool. So we talked about how the embryo destruction, embryonic stem cells, that is an issue. That's something that we're concerned about. And a Japanese researcher, he got around that ethical concern by creating something called an induced pluripotent stem cell. He took a little chunk of skin from someone's arm. I always do this, but no one ever does their arm. It's always their tush. No one wants a scar on their arm. So he took a little chunk of skin. He dissociated it into a layer of cells, and this is what that would look like. These are fibroblast cells, skin cells, and hopefully you can appreciate that they're kind of spindly, and they have a lot of cytoplasm. They kind of look like these big stars, and that makes sense within the context of their role within our body. They're here to create a barrier, and so they're going to stretch out until they find a friend, make some junctions, and then hang out there. So what this researcher discovered is that there are 4 genes that are sufficient to confusing these cells into thinking that they're embryonic stem cells. And this is just so cool because what it allows you to do, like I said, I don't have an issue that much with the embryo destruction given how infrequently it happens and the promise of these cells. But why this is so cool is because if I want to study Parkinson's disease with an embryonic stem cell line, let's say I differentiate it into neurons and I'm trying to figure, I put some genes in, the crux of the issue is that I don't know what kind of neurological development that, if that embryo had gone on to develop, would they have had schizophrenia? Would they have had Parkinson's? I don't know. And so I'm basically kind of studying an unknown within a way bigger unknown. And so at the time, that was all we could do, and so it was good. But what this allows us to do is to take patients with Parkinson's disease, so ones that we know have a genetic background that is both necessary and sufficient to get this disease, take little chunks of their skin, turn it back in time, and then differentiate into pluripotent stem cells or embryonic-like stem cells, and then differentiate it into neurons. And we do this in parallel with generally a sibling or a parent who is not affected by the disease. So genetically, they'll be somewhat similar, but they, for whatever reason, won't have developed the disease. And we do that in parallel, so that at the end, we have 2 plates of neurons, one from a patient with Parkinson's and one from a patient who doesn't, and we can ask ourselves, what's the difference? Let's tease apart, what is it that's making the Parkinson's disease cells more sensitive, maybe? Do we see them degenerating faster when we put on different environmental stressors? Because again, this is challenging because we're mimicking a disease that usually takes 60, 70 years to develop, and I had to graduate with my PhD at some point, and so and so we're trying to kind of mimic this a lifetime of injury, usually by adding stressful things to the cells and trying to find what we call a phenotype, a difference between the cells from patients with Parkinson's and the ones without. The whole purpose being to do drug screening, that we can then, once we find a phenotype, we can apply drugs to the cells, generally FDA already approved drugs, to see if any of those are capable of reversing that phenotype or preventing the phenotype. And then we believe that if we find a drug using this model, that it'll have a higher chance of success when we get to the FDA level, because we already know it's safe in humans, since it's already approved, and we can show that it works on human cells. So that's, yeah, we do this in the lab. I have chunks of butt brought down to my lab on a regular basis that look like this, and then when we reprogram them, this is kind of what the process looks like. So in the background here, hopefully you can see that there's all these fibroblast cells. They're much denser now because they've been growing there for weeks. And then we see this little colony kind of growing up in the middle, and people will sometimes say, well, how do you know it's working? And so, you just kind of know. And so what we do then is we pick these little colonies up, just the whole individual colony. This is maybe 200 to 400 cells. And then we put it on a plate, a new plate, without any of those fibroblasts in the background. And I don't know if you can tell the difference between this colony and the picture that I showed earlier, but I can't. If you give me a plate of induced pluripotent stem cells, the ones that are made from the skin, these inducing the state of pluripotency, if you give me a plate of those and a plate of embryonic stem cells, I couldn't tell you the difference. And that's really, I think that's really cool. He got the Nobel Prize for this work. It first came out, I think, in 2007, and he got the Nobel Prize in 2014, which is a 7-year turnaround, which is incredibly, incredibly impressive. Usually the Swedes take like 30 to 40 years before they determine that your work is worthy. So if after 7 years they said, okay, you got it, then it shows the impact of this research, I think. And
so what do you think is happening? So this is all really cool stuff that's happening now.
These are the neurons.
These are the neurons.
These are the neurons. These are the neurons that we make out of those embryonic stem cells from the patients. Mm-hmm. And I think they're just absolutely beautiful. What you can see is the cell bodies here, and then they project to the other cells. Everything circular there is where the nucleus is. And then along these tracks, that's where the synapses are. That's where the synapses, where the cells can actually communicate with each other. And when we look at these diseases, that's one of the areas that we're really looking at, is trying to see what's going on at the synaptic level.
And so what happens with the neurodegenerative diseases is that the synapses stop working correctly. They become disorganized and then they die.
Yeah, the cell, for a variety of reasons, will start to kind of retract and pull back its projections, and the synapses will, no longer be
functional. So what we're talking about is, so you make a bunch of new ones, right? and you replace those in the brain to go with the other healthy cells, and then everybody's healed, and we don't have Alzheimer's or Parkinson's anymore. But the way that you do that is quite a process. Talk about the process.
I'm sorry, the process of...
The trials that are going on right now with Parkinson's disease.
Yeah, absolutely. So that they're actually, as far as I know, with embryonic stem cells, there aren't any trials going on right now. There might be some with neural stem cells, but with the disease modeling, with what we're really trying to do is be able to find new drugs for these diseases, and our hope is not really to cure or even to treat, but to prevent. So what people don't realize is that with these diseases, Parkinson's, Alzheimer's, there's about a 10-year phase prior to the symptoms that we associate with the disease. So the tremors for Parkinson's or the memory loss for Alzheimer's. There's what they call for Parkinson's a non-motor phase, where patients will experience loss of smell, incredibly disturbing REM dreams, some autonomic dysfunction, meaning the things that you don't realize that your brain controls, like your heart rate and your blood pressure, that those things can become a little dysregulated. And so our hope is that we can talk to clinicians and have them keep an eye out for these collection of symptoms that otherwise might go kind of unnoticed if you think of them separately, and then combine that with some kind of biomarker blood test so that we can predict whether or not you're going to get Parkinson's, or basically tell you already have Parkinson's before you've even thought about shaking. And what we would then like to do is then come in with a compound that is neuroprotective and can save all the neurons you have. People don't realize you have to lose upwards of 60% of your dopaminergic neurons before you start having tremors. And so we'd like to catch the patients before we really get to that point.
The key is to catch things before they get to the symptoms.
Yeah, exactly.
So if somebody has this in their family, would it be wise for, if the mother or whatever, would it be wise for, and at what age does somebody come in and start to try to get?
Yeah, it's a great question. I get this question all the time. And if we had that drug that was neuroprotective that can prevent the disease, I'd say, yes, come in, get tested. Why don't we all just take that drug? I don't know. But the fact of the matter is we don't have that drug yet. And we don't, if you know that if you think you're going to get Parkinson's disease, there's really not a lot that you can do to prevent it outside of the things that we really all should be doing anyways. A diet rich in antioxidants, just leafy greens, anything that has, I told the kids this morning, anything that's bitter or sour is probably going to be good for you. Oranges, they have a ton of ascorbic acid in them. Blueberries, all these kind of foods. But that being said, you're not going to prevent your disease from that. It's a lifeline of things that might be able to make it better. Anti-inflammatories like ibuprofen have been shown to reduce the risk of Parkinson's disease. Some of those studies are controversial, but it seems like ibuprofen stands out compared to the other NSAIDs or non-steroidal anti-inflammatories. And so, but is it worth knowing an answer to a question that we don't understand? I don't know. People don't realize the most common gene for Parkinson's disease is LRRK2. So and if you have the LRRK2 mutation, you have about a 50% chance of getting the disease. So the most common disease gene that we know about for this disease isn't even really that predictive, and so it's challenging.
One thing she told me that kind of knocked my socks off was she was talking about her grandmother having Parkinson's. I was talking about my grandmother having Alzheimer's, and we're at the lab. Everybody else is gone, and I said, "Well, could you tell if you do your own skin sample if you're predisposed for Parkinson's?" And she said, "I could." And I said, "Could you tell if I'm predisposed for Alzheimer's?" And she said, "I could." I'm like, "Oh man, really? Do you want to know?" And so that comes up the whole question: do you want to know? And you really don't want your insurance company to know?
No, you don't. Yeah.
Do you want to know for planning purposes? You know, and how would you live differently? And I think that's a really big individual thing that we each need to think about. Do we want to know? If it's in our family, do we want to know? Because we can find out.
Yeah. And to maybe think, if we did know, how would that change? What would we do differently?
Right.
And that to me is the crux of the issue.
And it might be a way to better plan our ending.
Yeah,
If we have
absolutely.
an idea that we're gonna need a lot of care, then maybe we need to get another job. So we can have in-home health care, gather our friends.
Absolutely. Just make plans before we're not capable of making plans. Yeah.
So, okay, that's fascinating. I know that everyone's going to want to talk some more about that. as well. And one of the things too that Leah said, if somebody has Parkinson's, it's very good also to bike ride bike in your home.
There have been studies that show that riding a bike can improve motor symptoms, and to me it fits in that category of if you don't use it, you'll lose it, kind of, and that when things become hard for us, we tend to avoid them. But with bike riding, I'm not sure. I don't know if they actually compared it to different kinds of exercise, or if that's just an easier exercise for patients with Parkinson's to do, but that they will have increased motor control after biking. And it kind of makes sense. You say you have to do mental puzzles if you want to stay sharp. It's kind of the same thing with your motor skills. The more you reinforce something, the stronger it's going to be. The more you use the synapse, the lot stronger it will be, and the harder it will be to kill.
It makes sense.
Yeah, it's intuitive.
So going from there on to our organs, replacing our organs. If we have diseased organs, can we have surgery to take out the diseased part? Can you grow in a petri dish with our cells to create the better part of that and have surgery, and then we're all better? I know that when Leah and I were talking in the lab, now it was like 2 hours, and she said, I've got to tell you something really special. She goes, the future is really amazing. Everybody should have, when they're born, a little snippet taken out of their butt, cryogenically frozen so that we can grow our own organs later. It makes sense.
It does. It does. It's a little.
And the limitation is how much it's going to cost. And I said, well, let's talk about that.
Yeah, it's how much it's going to cost, and then how much time it's going to take. And to me, those things. I also am a little bit wary of care that will really only go to the highest bidder, so to speak, too. And so I like to think of a world where we can replace the organs that we have, but also in a way that we can do it so that it's kind of equitable and easily accessible to everyone and not quite as time consuming. And the way that I see potentially being able to do that is to be able to make a bank of stem cell lines, ones that maybe that cover every human being on this planet, not specifically targeted to one person, but just in the major histocompatibility complexes. That I've heard that an estimate that 100 lines would be sufficient to cover every human on this planet. And so if we think about that, we could have this bank of stem cell lines, and you could just figure out what your type is, and then if you need a new whatever, you can go, they can make your new whatever out of that one line that you are compatible with. And then we don't have to worry about, who can afford or have access to that kind of research. And to me, that's really the future, the really exciting future of this kind of medicine, is to be able to do personalized medicine, but in a way that's global.
So let's go back, though, to there's something that's really cool that's happening right now with 3D printing and
yeah,
stem cells.
Absolutely.
So when we're talking about those growing the organs, which we can grow, talk to us about 3D printing and how that works.
Yeah, well, the reason that we would even consider doing 3D printing is because the big challenge that we're dealing with right now is how to make complex structured organs. So when we think about replacement technology, being able to replace organs, certain parts of the body are easier than the others. There have been 3 clinical trials that have been approved in this country using cells that you made from embryonic stem cells into what you could call an organ, and then transplanted them back into the human body. The first one was a spinal cord injury trial. The second one was a retinal pigmented epithelial, a trial to try to cure macular degeneration, so a kind of blindness. And the third one is ongoing in San Diego right now to try to cure juvenile diabetes, so type 1 diabetes. And these, I'd like to tell you more about that, but we don't have time. So those trials, the reason that those ones have been the first going forward are twofold. One, because we can make those cells well. For some reason, we can make the pigmented epithelium of the eye really well. We can make the spinal cord stem cells well. So it's about what we can make in the lab, but then also about what's easy to put into the body. So the eye is actually a relatively accessible site. And then it's also about how structured the organ is. So the pancreas is a little blob, and it has all the cells are little round blobs in it, and their job is primarily enzyme secretion. So they're just here to kind of make some enzymes. And if you compare that to a heart or a lung, where like the lung, you have all these little bronchial, alveolar, little reaches going in, and if you don't have that structure, the lung won't function. It's the same in the kidney with the nephrons. You could say the same thing about the brain, about the heart. And so the company in San Diego, the way that they're doing their trial is they've taken the embryonic stem cells, differentiated them into pancreatic cells, and then put them in a little device, a little capsule that has a mesh on either side, so and that's what they've actually transplanted into the patients, kind of in the hip, thigh, butt area. And the reason that's so cool is because the cells can't get out of that mesh. The pore size is such that the cells actually can't escape, which is important because we talked about the unlimited cell growth potential of stem cells. And so if we go from juvenile diabetes to pancreatic cancer, we've definitely done the opposite of no harm. And so this company, the mesh, allows the cells to stay in there. And so if a patient has any kind of problem with it, we can just take it right out. And the mesh is big enough so that the blood vessels can grow in, which deliver the glucose and the oxygen and all the stuff needed for survival, and most importantly, brings the insulin out. So that works. We just made a little mini organ and a credit card, and it's okay because there doesn't need to be any kind of structure to allow for its function. But there's a lot of organs where that's not the case, and that's where 3D printing comes in. What we're working on as a field now is how do we create these complex organs for transplantation so they're still functional. And there's a company in San Diego, it's called Organovo, and they do tissue printing. And so it's kind of like your inkjet printer, but instead of the cartridges for different colors, you have different cell types. And so let's say that you want to build an intestine. The intestine has these kind of intestinal crypts that have 4 stem cells here, 2 progenitors here, and then the intestinal cells there. And so they can actually put those 3 cells into their printer and then say, print 4 stem cells, 2 here, and then up in a crypt and down. And it's just so cool to see. They have videos of them actually just dropping the cells, and then the cells can have a level of organization that we can just orchestrate like that. It's just very cool technology. Another way that we're doing it, another way that we've been trying to get this structure is by decellularizing organs or making matrices that are. So if you decellularize, for example, at MIT, they did a heart. I think it was MIT, where they took a rat heart and they cleaned all the cells away using a detergent, and all that was left was the extracellular matrix that the cells had secreted, and so it kind of looked like this lace mesh. It was actually quite beautiful, and they seeded cardiac stem cells on it. And the cardiac stem cells grew around the mesh into a functioning heart that they transplanted into another rat and had it still work. So it's this. We're trying now to think about how we can build these complex structures using things like printing or lattices or just technology like that.
What's going on?
Yeah.
One thing I wanted to mention is that a lot of people are not aware of clinical trials that are happening and
Right.
how to get into them. So.
Absolutely.
You know this.
There's so many clinical trials going on right now, and our government has them all organized into a website called clinicaltrials.gov, shockingly. And if you go in and you type in stem cell, you're going to get hundreds of things. Most of those are on adult stem cells, neural stem cells or mesenchymal stem cells. But if you or anyone in your family could be interested in participating in these trials, I encourage you to go to this website and to look it up. And also, I'd like to, before we break, to say something about the quackery that is out there, because there are so many people promising stem cell cures, stem cell this, stem cell that, and I just urge you to be aware and to really think about whether or not these promises are founded in reality, because more often than not, they're just scams to make money, and they're preying on people that are having a very hard time in their disease state, and it's disgusting.
So there's most likely, if you see something that says have a free lunch and come in and learn how to use stem cells for your ligaments or your knee, so you don't have to have any knee surgery or hip surgery, and then you'll get $1,500 off your treatment, it's probably not.
That's probably not real. Yeah. And just when in doubt, call them and ask them, where is your work published? Where is the clinical trial? How do we know this is real? Show me your results. That's all you have to do.
Yeah.
And when in doubt, just email me. I'll look into it for you. Figure it out.
And we're going to do one last question before we have, I know we'll have a lot of answers, of questions for you. I mean, you touched on this a little bit. If you had unlimited resources, the lady's brilliant, and could see the future of what do we have to look for stem cell future? Mm-hmm. What do you see can be our solution, our answers, and things for hope for stem cells technology?
Well, I feel that we're at the most exciting point in stem cell research so far, because it's a whole new frontier of medicine. We used to, we treat people with compounds, usually drugs, and if you have a hard time with that, we just stop taking the drug. But with cells and with genes, with these kinds of therapies, it really is a whole new world of treatment paradigms where we have to be really careful that we're doing it right and that we don't cause anyone undue pain or cancer or anything like that. And so I guess my dream for the future would be that we are putting cells in people's bodies safely and really harnessing the potential and all the work that's gone into this field. And not only that we're doing it safely, but that we're doing it equitably, that we're being able to do it for people all over the planet as much as we can. And I think that the key to that really is making the bank that we touched on earlier,
This is what I want to go through.
where we can really have 100 stem cell lines that because when people do matching, for transplants, you go family first. You try to get someone who's as close to you as possible, because the likelihood of being a match with your immune cells is going to be much higher. And so if we can create a bank that has all of the major immune types recognized or represented, that we would then be able to use as our source for transplantation. And to me, that's the future. That's the full potential of stem cells realized.
And so we would have donors that would be able to donate, and they would fall into one of the 100 categories. And anybody from whether they're rich or poor can access that and have hope for the future for getting rid of their disease.
Yeah.
Amen, sister. I don't know if we have any roaming mics.
There you are.
Yeah. Do we have roaming mics or not? Okay. Okay.
Can you tell me where we're at with lumbar disc replacement and regrowth?
I think that do you have a herniated disc or never mind, you don't have to answer that for everyone else. Okay, so the lumbar, the discs are kind of this like jelly material that I honestly think that if we regenerate, it's going to be more through biomaterials and that kind of making kind of a synthetic material that will allow us that we will then inject in there. Likely not with stem cells themselves, if I had to guess. I have a herniated disc and severe sciatica, and so my fingers are crossed for that. Yeah.
Hey, that was a really interesting talk. Thank you. And I'm wondering if you are looking at senescence-associated secretory proteins or anything, that's kind of the opposite of the stem cell, because it feels to me like that's where the answer is. If you can find those in a stem cell, then you can think about tissue longevity and right. So I'm just wondering if you were talking about extracellular matrix and all of that, and I'm just wondering if you're looking at SASPs in any of your tissues.
No, and I've never heard of SASPs before today.
We should talk.
We should talk.
Oh my God.
We should talk.
We should talk.
Yeah, and then I think it's a great idea to look into different tissue types and see how we can get there. Yeah.
Here, I'm curious how cord blood, which is advertised for new babies, is that the same or different than you were talking about saving cryogenic samples? If you could explain that.
Yeah, absolutely. And I get this question a lot because we're everyone who's going through that phase of their life wants to know if it's worth it. You want to do the best thing for your baby as possible. And the answer is, remember that epigenetic map? Cord blood is kind of the closest thing to the top that we can get in a way, because it's the kind of the freshest, most stem-like cell. And so if we wait later, the stem cells that we'd find in our bone marrow, they're kind of a little bit further down. And so we're saving them a little bit with the promise, the hope that there will be something that we can do with them. If your child does go on to develop blood diseases, then they will absolutely be useful. But we started banking them before we even knew about these induced pluripotent stem cells. They would be a great source to make induced pluripotent stem cells out of. And so we're banking them knowing that they'll be able to be useful if the child goes on to develop any kind of blood-related diseases, but also with the potential that these cells might carry in future cures that we might be able to do, because, like I said, they're so fresh and but kind of high up on that hierarchical tree.
You talked about animals having stem cells. Do plants also have stem cells?
Yes, plants also have stem cells, and I wish I knew more about this, but I really like to garden. I have a very big succulent garden at home, and I love succulents because I think they're very stem cell-like. You can just break off a leaf, and then the cells that were around that at that node, they'll appreciate the signals of, decapitation, and those cells will kind of revert back into a stem cell-like fate and then become root cells. And they'll start making roots, and that's the property of plant stem cells. And I don't know nearly as much about them as I want to, but yeah, it's a great question. Great question.
So I've got two questions for you. Hopefully that's okay. You mentioned at the beginning you look for patterns when you make these synapses in these stem cells. I'm curious what role machine learning, humans are okay at recognizing patterns, computers are much better at it than us.
Yeah.
So I'm curious how important machine learning is in your field. And then my second question has to do with how you and your peers are funded and whether we have the right funding model. I look at what's happening like with internet startups, and the reality is we have thousands of people trying all kinds of things, the vast majority of which are going to fail, but it's a very quick way to find new things that are going to be really successful.
Right, right. Well, the first question was about pattern recognition and finding patterns in our cells, and whether or not we can use machine-associated assisted learning, and the answer is yes. In our neuro labs, we do a lot of that, and as we're scaling up in our studies, that's becoming more and more important, because so for an example, there's some cells, neurons will grow on plates that have electrodes built into the plate, and the electrodes will actually kind of poke up into the neurons, and they can map the neurons communicating with each other on this entire well level. Because usually when we look at how neurons communicate, we'll stick an electrode in one, an electrode in another, and you kind of just have this paired reading. But in these plates, we can now look at hundreds of cells and how they're communicating with each other, and then do that 100 times over in the plate and all the different wells. But to be able to analyze that data and to be able to find patterns in that, we really do need to be able to use high-powered computers for pattern recognitions, again, to find those phenotypes, to find differences between the cells from a schizophrenia patient or another one, or patient without that disease. And then to the second question about funding, funding has definitely changed in biomedical research in the past 10, 20 years, and it's shifted from being mostly federally funded. The SALC used to be in the 60%-ish federally funded. Now we're getting down into the 40s. We're not funding science. We're not prioritizing science as a country. We're prioritizing war. If you look at our war budget and how that's increased, that's where our NIH budget went, and we just again have to think about these things as a society and where we want our money to go. And I'm actually very lucky being in the stem cell field because of the regulations that Bush put on the funding. It meant that it really invigorated a lot of private donors and foundations to get involved in the science. And I'm telling you, it's wonderful because people are actually getting more involved and funding things. And people come visit my lab, so it's nice to have that too. And that is now how the SALC is mostly funded, through private foundations and fellowships, including we have a huge grant that supports my core entirely from the Helmsley Foundation, which is, I don't know if anyone has heard about Leah Helmsley. She's the one that...
I thought it went to her dog.
She left her millions to her dog, right? Dog, yeah. And then the dog was really generous, and the dog was like, look, get me a couple Kongs, and so it's kind of like what they did with the Howard Hughes Medical Foundation. Howard Hughes died with tons of money and no real plan, and so they put that into a medical fund, and so they did the same thing for Leah Helmsley after the pooch was taken care of. And so we got a $43 million grant from them, which is beyond generous, and that will fund my core through 2017, which is just so nice. That's the way you should have to do science. You shouldn't have to worry about, we're really appreciative of that. I do wish there was another funding model for science that kind of got it, that was a little bit more directed, because science fails all the time and our funding is not good. We are usually using the money from a project to start another project so that then we have preliminary data to apply for a grant. If that makes sense. It's kind of like the money is always one step ahead of it, so to speak. And that's not ideal. And so I wish that there was kind of a different model where you could kind of do this more shotgun approach, expecting failure, but also just keeping an eye out for the stuff that stands out.
Hi, it's always nice to see the person asking you the question.
It is. I was going to say thank you for standing up.
Right, welcome. On the subject of pluripotency and the scale that you've been talking about. I'm curious about adipose tissue, which is a lot more accessible for pretty much everybody than bone marrow, say,
Yep.
right, is one source. And then also, I'd understood that the original, is it the blastocyst, the original creature, whatever it is, the organism, whatever you would call it, within the first 14 days or something like that, or 30 days or something like that, if you remove some cells from it, they will grow back, correct?
Yeah.
So you want to really help your kid out?
Yeah.
Take that little growing thing and take some cells out of that, which are true pluripotent stem cells, right? Do you have any comment on any of this?
Yeah, I mean, yes, I do. What was the first question again?
It was
Oh yeah, adipose tissue. I don't know as much about that. There is a lab in Massachusetts working on that. Chad Cowan's lab, I think, is doing adipose tissue, COWAN, and I'm not sure if they reprogrammed from it yet. But what we're trying to work towards is being able to reprogram blood cells, or even just like a cheek swab or something like that. We've been able to do hair follicle, so you can just pull a hair out and reprogram those cells. So that's kind of a less invasive way to do it. And then in terms of the true pluripotency, yeah, what you're talking about I think is very, very cool. We talked about it with the high school kids, but in those first couple of divisions, you can go from the 1 cell to the 2 cell to the 4 cell. Starting at the 8 cell stage, you can take one of those cells out, and the developing blastocyst will continue to develop and be fine. And the reason that we do this is usually to diagnose for genetic diseases. For example, Huntington's disease. If you and your mate, if you have Huntington's disease or a history of Huntington's, I think it's your ethical obligation to get IVF and to do this pre-genetic diagnosis, so that you only implant babies that have blastocysts that have the potential to not develop the disease, that don't have the repeats. Huntington is a disease of glutamate expansion, and so you want to make sure that you're not implanting, children that are going to have a hard time at life. And so that's.
Yeah, I don't think a lot of people know that. We talked about this today, and Leah said, I don't know how anybody with Huntington's disease cannot do this, because you can totally prevent your children from having this.
Yeah, Huntington's at this point is now 100% preventable, unless it's a de novo case, and those are very, very, very rare. De novo just meaning new. And so I think that if you are I honestly agree that if you're going through IVF, take one of those cells out. But I also think that a lot of people that are going through IVF, they don't really want to disrupt the doctor, they don't really want to disrupt the process, because generally they're there for a reason. And so they're not just going to start taking cells out just to have that potential future, since we can do the reprogramming later in life. But, I kind of agree. If you know it's not going to hurt, why not? Just kind of divide that up in two, turn one into a stem cell line, and then have it banked.
Where do you have it banked? How do you have it banked?
So there's companies that are centered around this, like the cord blood. There's cord blood companies that are associated pretty much with every maternity ward. And then a lot of these IVF clinics do cryopreservation for a fee. And then it's not only an initial fee, but then an annual fee for maintenance.
FedEx and then the annual fee.
Exactly.
One last question?
Everybody usually talks about the neuronal systems in the CNS and your Parkinson's, but what about in this deterioration of these specific neural cells? What about the role glial cells play in that?
That's a great question. That's what I did my PhD on. The glia are playing a huge role in these neurological diseases that has gone otherwise unnoticed for a very long time, and I think that there's starting to be more of a focus on glia. So just to take a step back, there's a couple different cell types in your brain. You probably have heard of like the white matter and the gray matter. Those kind of represent neurons and glia, and glia is Greek for glue, because we always kind of thought of them, it's the most common cell type in the brain, and they thought that it kind of held the brain together. But still, when we think about neurological disease, we focus very much on the neuron, and it kind of makes sense, because the symptoms that patients experience are from a result of a loss of neuron. But when we think about treatment, I think that we really need to be thinking about the whole brain. And glia can play a role in two ways. They're support for the neurons. And so if the glia are having any problems, the neurons aren't going to be well supported, and they're going to be more likely to degenerate. Additionally, if we boost up the glia, maybe that means that there'll be better support. They're going to release more, BDNF and all these other factors that they secrete to really bolster our neurons. So there's that one aspect of glia providing trophic support for our neurons. And the other aspect is inflammation. glia are amplifiers of inflammation. Microglia are the initial responders of inflammation in the brain, and astrocytes are the most common kind of glia. They can really amplify this inflammatory signal. And I believe that chronic inflammation can actually be contributing to the progression of these diseases, and is something that we should that we need to start looking at more. And so for my thesis, I did a screen and I found a compound that I think will be a very good clinical candidate for Parkinson's disease because it is both neuroprotective, it can increase the transcription of genes, it can make the cell make more of the genes that are really, really important for neuroprotection, so it makes the cells stronger, but it also dampens inflammation in the glia. And so it's kind of this two-pronged approach, but it's a natural product, and so we can't patent it, so they're not excited about taking it forward. Welcome to science.
So
Do you know what we
before
forgot?
Mo- bef- huh?
Do you know what we forgot?
What?
The one thing you asked me to bring.
The heart.
The heart.
Okay, oh, we have one really, really cool slide. This is when I saw this in the Petri dish, I said, okay, you're coming to FiRe.
Yeah, when we talked about what slides, she was like, oh,
Yeah.
you just make the heart cells. So you differentiate the cells based on what disease you're studying, right? And so since I was studying Parkinson's, we turn these cells into neurons. But one of the biggest feathers in my lab's hat is a project we did with UCSD, over 200 patients with cardiac diseases, including one called long QT syndrome, where you are more likely to have a heart attack because your Q and your T interval, that ratio is different. And so what we do with those cells is we turn them back into time, and then instead of turning them into neurons, we turn them into heart cells. And then we try to see if we see a difference. And this is what we see. So what we're looking at here is a whole web of cells. So this is all these little tiny things in here are cells, and what's so cool about this is that they're beating in this coordinated fashion that has kind of a da-dun nature to it, this kind of biphasic nature. When I was at Harvard, I was making blood. We were trying to make blood cells to cure leukemias in children, and blood is a very similar lineage to this. And 22-year-old Leah sitting at the microscope, like trying to look for some red cells, and then I see dun. I ran into my boss's office, like, I don't know where life begins. I don't know what we're allowed to do, but I need you to come look at something. And then after that, actually, every donor that ever visited Children's would come by our lab because these are real crowd pleasers. Yeah. And this is where I work. This is where I'm lucky enough to work.
Yeah.
We had Chihuly come and visit and decorate our place for our 60th anniversary with his glasswork. And right there's the Pacific Ocean. That's where I hike down on a weekly basis to go swimming. And I just feel like the luckiest person in the world, not only because of what I do, but where I get to do it. So, anyways.
Is Edwin Hunter in our office in our group tonight? Okay. Well, we have to give a huge thanks to Edwin Hunter, who allowed us to be able to meet Leah. And thank you for coming tonight. And thank you to Park City for joining us for all of these seasons. I mean, this is so much fun for me, and it's so cool to be able to bring people that we meet from out there into our world here. And if anybody would like to go to FiRe, go to futureinreview.com, and you'll meet many more speakers that we'll choose from for our next series for next year. And thank you to Park City Institute. Mo, you are amazing.
Thank you.
Being partners with these people is like nothing else. So,
Thank you. Thank you.